Gene Editing and Human Enhancement number 3, 2010 to the present time. The CRISPR Revolution. I'm Ted Peters and my colleague Arvin Gouw. Between the two of us, we'll talk about the science, the theology, and the ethics of the CRISPR revolution. The analysis of the CRISPR revolution that Arvin and I plan on providing here is an exercise in public theology. Public theology is conceived in the church, reflected on in the academy, and addressed to the wider culture. We have 5 tasks: the pastoral, apologetic, political, prophetic, and the scientific, to develop a theology of nature. Arvin is a practicing laboratory research scientist who engages in theological and ethical reflection. Fasten those seatbelts. We've got a fascinating ride coming up. Here is something you do not need to remember. Our word CRISPR is actually an acrostic for clustered, regularly interspaced, short, palindromic repeats. These are repeating sequences of nucleotides in the chain of nucleotides that make up your and my chromosomes. Originally, in evolutionary history, these short palindromic repeats would grab invading viruses, but that's not relevant. You can ignore that. What is relevant is that these very specific patterns, the clustered, regularly interspaced, short palindromic repeats, sit there in your and my DNA and appear to present precise targets. If we want to shoot an arrow, and the arrow would be the enzyme Cas9, we can hit the target if we aim at the CRISPR. So think back of those dreams of the 1990s during the Human Genome Project, or the dreams during the growth of the stem cell industry. What we get with CRISPR is a practical technology which makes it possible with relative simplicity to alter the human genome. Gene editing. Is gene editing ethically controversial? You bet. Everyone applauds Emmanuel Carpentier and Jennifer Doudna, the 2 women who gave us CRISPR, and both of whom won the Nobel Prize for their advance in science. Everyone applauds the science, but we worry about society. What will happen if we have free market gene editing? And someone's going to want to enhance the human being through this technology, right? Human enhancements, unbridled capitalism, inability to plan for the future. What kind of a mix that fills us with trepidation is that? In 2019, my colleague Arvin Gouw and I planned and led a conference on the science of CRISPR gene editing and its potential impact on the future of the human race. Sponsored by IRAS, the Institute on Religion in an Age of Science, at their annual conference held at Star Island off the coast of Portsmouth, New Hampshire. Here on the left with the microphone, you can see Michael Roos. He's a well-known philosopher of biology. And to his left is Arvin. And to Arvin's left, with the Lake George sweatshirt, is myself. Second from the right is Lisa Fulham. You might know Lisa from the GTU. Well, we're going to turn to Arvin now and have him give us the ABCs of CRISPR science.
Hi, my name is Arvin Gouw, and this is Danielle Fumagali. In the picture here, my wife. I am a scientist by training with some theological training as well. My main research involves cancer research, more specifically cancer metabolism. CRISPR is not my specific area of focus in terms of developing novel CRISPR technologies. However, as many other scientists, CRISPR is definitely one of the techniques that are very important in cancer research and many other fields of research. So basically, CRISPR screens is a way to find novel genes that would be amenable to be targeted for therapeutic development using CRISPR screens. In other words, this is knocking out genes one by one to figure out which gene knockout will cause cancer death is really an agnostic way of discovering novel therapeutic targets. So CRISPR screens in the context of various kinds of cancers and various kinds of diseases really is one of the ways that we discover new targets without presupposing certain hypotheses and thoughts that could be biased to begin with. So this is how the research involves. We discover that cancer metabolism is one of the pathways that are very important for cancer cells to survive, such that if you knock out or inhibit the functions of these metabolic genes, then the cancers actually regress. So in short, this is the kind of work that I do as a scientist. This talk will be composed of 4 parts: CRISPR mechanism, CRISPR applications, CRISPR dangers, and CRISPR regulations. First, in terms of mechanism, how does CRISPR work? It's originally discovered as a bacterial immune system. So if we think of a bacteria as a single cell organism, how does it protect itself against viral infections? So upon the entry of a certain virus, the virus will release insert its genetic material to the bacteria. The bacteria will then chop off a piece of that genetic material, incorporate it, and form what is called a CRISPR RNA. Upon the second time of viral infection, the CRISPR RNA of the bacteria, should it match with the second infection, it'll cleave that viral RNA. So this is the basic system of a CRISPR bacterial immune system. Basically, as scientists, it was very an ingenious discovery by Jennifer Doudna and by Elizabeth Charpentier that they can change the guide sequence of the CRISPR RNA to match with any gene of interest. Therefore, you can basically delete or knock out any gene that you want. Right? So you put a guide sequence for gene X. When that sequence matches with gene X, then Cas9 enzyme will cleave that gene of interest. For that, Jennifer won the Nobel Prize with her colleague. What are the applications of this technology? There are many applications of this technology. It's been used for multiple species since its discovery in 2012. For example, for E. coli, E. coli can be CRISPR to consume carbon dioxide to produce ethanol. Algae can be CRISPR to produce biofuel. So these are two inventions that are very good to address global warming problem. CRISPR in agriculture, plants have been CRISPR to allow better nutrition, longer shelf life, they have some disease resistance, drought tolerance, and higher yields. So there are various functions of CRISPR here in different organisms. In animals, this is the first dog that was CRISPRed. The dog is named Hercules because the dog that are CRISPRed became very buff, much higher muscular density. Pigs have been CRISPRed to be petite, and they're called micro pigs. They're sold as pets for $1,500 from China. Eggs can be CRISPRed so that they're no longer allergenic. Mosquitoes can be CRISPRed so that they can not spread malaria anymore. So let's pick up this example of the muscular density manipulation, myostatin deletion. It leads to increased muscle mass. We can imagine two possible scenarios. On the left, for a Duchenne's patient, you can delete the myostatin using CRISPR, and then the patient will then gain some muscle mass. That's good therapy. On the other hand, for soldiers, you can imagine Captain America here, a normal person being CRISPRed, and then becomes very muscular. That, I think, would be considered enhancement. So this is an example of a single CRISPR application that can be used for both therapy and enhancement. Organ transplantation. Pigs have been used as a farming tool for various organs. The current problem is that when you receive donors from other individuals or other animals, there is a host versus graft rejection. Right? So in order to avoid that, you can CRISPR all the immune markers that causes us to reject these organs. So this has great potential in terms of organ transplantation and organ farming. CRISPR gene drives is the case where you can CRISPR a certain gene of interest and allow it to copy itself to the other allele. All right, so let's look at the first top diagram here. You have altered gene only in one copy. So if you have an altered gene in one mosquito mating with another mosquito wild type, only 50% of the progenies will carry the altered gene. However, in the bottom panel here, upon gene drive, 100% of the progenies will have the altered gene because basically the altered gene will copy itself to the other allele. So you have 100% penetrance or 100% chance of passing it on to the next generation. Okay, so you can imagine that if you insert a gene that causes lethality in a certain species in a mosquito, you can actually drive the whole species into extinction. How can genetic mouse models help us? CRISPR genetic mouse models are very easy to use, very efficient, and they can recapitulate a certain patient's genetic makeup. And we can study the mouse model before applying therapy to the patient. So this is, we published a book here applying next generation sequencing and transgenic mouse models for rare disease research, where we are able to recapitulate a rare disease patient's disease in a mouse model to discover and test new therapies. Why is this good? Right now, there are 4,000 monogenic diseases. Monogenic diseases means diseases that are caused by a single gene only. If we're thinking about a person's height that is caused by multiple diseases, it's regulated by many, not diseases, but regulated by many genes. But there are certain diseases where you know a single mutation in a certain gene will definitely exhibit a pathological phenotype. So in that sense, these 4,000 monogenic diseases are the easiest targets. These are the low-hanging fruits for CRISPR therapy. And this is what I propose in the book Clinical Genomics and Rare Disease Research, published by the Royal Society of Chemistry. So how do human CRISPR gene therapy work? Well, there are two options. You can have ex vivo gene therapy where you pick up someone's somatic stem cells or somatic cells, and then you CRISPR it. Once you CRISPR it, then you put it back in. That's why it's called ex vivo. It's outside of the body. However, you can also have in vivo gene therapy where you basically insert the CRISPR, and the CRISPR in the body will reach its target organs and then do the CRISPR in vivo inside the body. So these in general are the two different methods of applying CRISPR gene therapy. CRISPR gene therapy is very, very popular right now because there's also a financial incentive. There is an Orphan Drug Act in the United States where if a disease does not have treatment currently, if you discover a treatment, then you have a possibility of accelerated FDA approval, but definitely some kind of IP protection law allowing protection or monopoly of that invention. The first CRISPR human trial started in November 2018. Jennifer herself was there in Europe when this began. This was a landmark point. Now, what are the CRISPR dangers? What are the dangers of doing CRISPR therapy? There are basically four basic ones. First one is off-target effects, meaning you're not sure if you insert a certain CRISPR knockout single-guide RNA that it'll target the specific gene of interest. There are 3 billion base pairs in the DNA. Your single guides is probably 20-something sequences. How can you make sure that these 20 pairs are specific enough to a specific gene amongst the 3 billion? Right? Mathematically speaking, we know that this is pretty precise, but there is always possibility of off-target, just like in any treatment. Second is on-target effects. Suppose you target that gene X. We don't know the unintended consequences. A classic example is sickle cell anemia. Sickle cell anemia actually confers resistance to malaria. Right? So if you cure sickle cell anemia, you might get malaria, for example. Chimerism is where not all genes get edited, leading to differential survival of cell subpopulations. So this is creating a chimeric organs where the untreated ones will probably overdrive the treated CRISPR, rendering them useless. Epigenetic factors is a much complicated mechanism where epigenetic means something that is outside of the gene. So just because you have the gene CRISPRed out or CRISPRed correctly or CRISPRed inserted correctly does not mean that it will be expressed. So and a classic example of this, for example, someone might have a gene that causes cancer, but it doesn't mean that the person will get cancer because the gene is actually wrapped up in a certain histone configuration, repressing the transcription of that gene. So therefore, this is called epigenetic repression. Just because you have the gene doesn't mean that it'll be activated or that it'll work. You can imagine you can put in CRISPR in a certain gene, but it is not expressed. So therefore, your CRISPR modification is rendered useless. All right, there are also scary applications of CRISPR. You can imagine next-gen bioterrorism. Obama advisors urge action against CRISPR bioterror threat. There's an episode here of Designated Survivor where someone designed a CRISPR agent that targets people with only dark pigmentation, meaning only people of color. Right? So there are many possibilities of the CRISPR applications for bioterrorism. There are medical applications combining CRISPR with nanotechnology. You can imagine putting CRISPR into some kind of magnetic nanoparticle, which then could be dragged into a certain area using magnetic force for treatment. The worst nightmare happened with He Jiankui when he first CRISPRed the first twin babies, Lulu and Nana, supposedly knocking out a gene called CCR5. CCR5 is a gene that is required for HIV to latch onto our cells to infect. So he was hoping to create HIV-resistant babies when he knocked out CCR5 using CRISPR. So in the idea behind CRISPR designing babies is also interesting. The language itself, the designer. Design implies that genetic modification X will cause a change X. However, as we talked about, there are unintended consequences and there are epigenetic effects. There's a certain genetic determinism here, or genocentrism here, that happens in the language when we talk about CRISPR designer babies that we need to be really aware of. The language itself carries a lot of weight, especially in public media. Now, with regards to CCR5, he received a lot of criticism because CCR5 deficient mice, for example, physiologically, can make the mice more susceptible to genital herpes simplex virus type 2, meaning that CCR5 has normal physiological function, such that if you knock it out, you might be resistant to HIV, but you lose the normal physiological function of what CCR5 is supposed to do. Okay? Then other studies came up. CRISPR twins might have shortened lives, according to some studies. Another study shows, though, the opposite effect. CCR5 is a suppressor of cortical plasticity, meaning CCR5 suppresses the ability of neurons to arrange itself and the process of learning and memory. So supposedly then if you knock out CCR5, you alleviate or you inhibit the suppression of cortical plasticity and affect making the baby smarter. Okay. So we don't know what will happen to these twins. But you might beg the question, well, if we know the genetics and we know all the epigenetic and environmental factors behind the regulations of certain genes, can't we control, actually design the ideal baby? And this is what one would call a biological determinism, or biological plus environmental determinism. And this is somewhat the approach or the spirit that's running behind behavioral genetics, sociobiology, and more recently evolutionary psychology. Right? And if this were true, that your genes plus your environment and lifestyle becomes who you are we then truly free? Or simply, we are unconscious of the gene environment predetermination that drives us. Finally, CRISPR regulation. The problem with CRISPR is not like other technologies where it is difficult to access. We have the opposite problem. There are too many people who can access this. For example, CRISPR can be taught to middle school kids without a problem. Here, biohackers are using CRISPR to inject himself. CRISPR kit, if you go to Amazon, is $170. All you need are water and microwave. That's pretty cheap, even though it might not be prime shipping. But if you're patient, you can get this kit for $170. It's for bacterial genome engineering, but one can imagine once you learn the skills, editing a bacteria to create a superbug is not funny either. What are the regulations? ACMG says raises the issues of the side effects that we talked about, off-target effects, epigenetic consequences, and so forth and so on. And the National Academy of Sciences also raised the four major concerns that I talked about. Being well aware of the significant advantages of CRISPR, and in general, all those bodies of societies, they basically recommend CRISPR for therapy, but not for enhancement, for somatic, but not for germline modification. So these are the several themes that we need to consider. There are certain lines that we need to consider. Natural versus modified, external versus internal, therapy versus enhancement, somatic versus germline, and permanent versus reversible, which is what I'm kind of pushing forward recently. Are we playing God? This is always the question. This is the title of Ted's very popular book, Playing God. Should we alter nature? Is this a Promethean, Frankensteinian hubris, according to Cass? There's a reason why nature is the way it is. Do not tinker with it, says Rifkin. However, one can counter-argue, is nature how things should be? This is an is-ought problem that was presented by Hume. We are playing God. Well, maybe we're playing with God if we use Hefner's language of created co-creators. Nature is incomplete, according to Deacon, and its fulfillment will be made in the future, according to Ted Peters. So maybe it is okay to do this. There's a blurry distinction as well, scientifically, between revertant mutations versus CRISPR editing. You can imagine a case here of Wiskott-Aldrich syndrome, WAS, where a wild-type genotype of GC, when mutated becomes AT, is very pathological, but a significant percentage, about 10% or so, has a second revertant mutation that reverts it back to GC, which is the wild-type mutation, well, the wild-type genotype. Does this not mean that CRISPR is basically helping Mother Nature cure herself? So, natural versus artificial, I think this is a blurry distinction. Second, external versus internal. We seem to be more bothered if something is modified internally. When we wear glasses to fix our eyesight, that's less of a problem. When you have a brain transplant or retina transplant, that becomes more concerning. But when we modify the brain to be able to see things that we normally cannot see, then we really have an issue about the moral implications of such modifications. The same thing with Adderall. Students often abuse Adderall to be able to allow to focus. But if we were to CRISPR the frontal brain to allow us to focus better, to be smarter, so to speak, then we have a more critical problem. So there is something about internality that makes us disturbed when it comes to CRISPR modifications. And I guess the more fancier question, or the theological question, is where do we locate our essence, our humanity? Are we our genes? Should we alter our DNA? Identical twins have the same set of genes, but they're different persons. So maybe we are not our genes. Maybe we shouldn't be too concerned about CRISPR modifications. If so, are we walking brains then? Should we not genetically alter our neurons because that'll change who we are? To get to a higher level, are we nodes within social infrastructure that our brains are situated in certain social structures, and therefore it's the social structures that predetermine who we are and define who we are? But we can expand this, go up higher one level, and say, are we how we respond to God? Therefore, we are independent or free even from the social structures that we are embedded in. Another theme that
Ted here on human enhancement. Elsewhere, Arvin argues that we really cannot draw a sharp line between therapy and enhancement. Ethically, the issue really is enhancement. Here are his working definitions. Genetic therapy refers to the manipulations of the genome to treat individuals or their progeny with known diseases, disabilities, or impairments to restore them to a normal state of health. Enhancement refers to the use of genetic alteration, pharmaceuticals, devices, or other means to alter the normal workings of the human body or psyche, or to be better than what is normal and native to healthy physiology. This distinction has become the place where most have drawn the red line. Bioethicists routinely affirm gene editing for therapeutic purposes, but they reject gene editing for enhancement purposes. And even though that's commonly done in bioethics, Arvin is going to say, well, the line isn't as sharp and red as most people think it ought to be.
Consider is therapy versus enhancement. The fact that the guideline says CRISPR is okay for therapy but not for enhancement assumes that there is a normal line, such that anything below normal is receiving therapy, anything above normal is receiving enhancement. But is this the case? One can imagine if you provide therapy to everybody below the average, then the average will move up, such that the person who were higher than average before now are actually below average and would indeed be receiving therapy instead of enhancement. So there is a moving line here, and this is what I propose in the Palgrave Handbook of Philosophy and Public Policy. Another example of this normalcy change is the condition of astronauts. For us humans on Earth, it would be enhancement to be resistant to hypoxia and radiation. However, for astronauts, that's actually necessary. It's a preventive medical treatment so that they can do their job. Right? So again, the term normal needs to be reevaluated in creating these various guidelines. You can read more about this from Human Enhancements for Space Missions. Should we enhance our children? Will we be blamed if we edited their genomes? Will we be blamed if we did not edit their genomes? If the technology is available, we are responsible to decide. To not act is to have decided. If we do decide to use CRISPR to help edit the genes of our kids, will this lead to some kind of a commodification of children? The real question is it truly for the baby's benefit to be edited or for the pride of the parents? Have parents commodified their babies? What if the gene editing goes wrong? Do we get discounts? What if parents keep trying to have new babies just so that you can end up having the perfect baby? And at what point is that baby no longer our baby because it's genetically so different from the parents?
Already during the Human Genome Project days, bioethicists drew a sharp moral line between somatic cell therapy on the one hand and germline modification on the other. The somatic cells are the ones that already exist in your and my body, and everybody agrees that if gene editing could help cure us from monogenic or other diseases, we ought to proceed post haste. On the other hand, if we edit the human germline, the gametes that would influence every generation to follow us for the foreseeable future, and because we cannot anticipate the side effects, it's a big no-no. Don't edit the human germline. And here you can see a number of scientists in the wake of the advance of CRISPR gene editing are saying, let's use gene editing, but not engage in germline intervention. That is currently the widespread consensus.
Another thing to consider, germline versus somatic. Right now, germline mutation is very frowned upon because this is the kind of mutation where you temper with the gametes themselves, such that the whole body or every cell in the body contains this specific CRISPR modification, and moreover, your gametes will also have those CRISPR modifications. So this is the kind of modification that impacts all future generations. Somatic modifications, on the other hand, is you modify certain cells in the body only, maybe the liver cells only or the muscle cells only, and it's not global and it doesn't go through the next generations. There are germline therapy arguments, some pros and cons as explained by Eric Jungst. Medical utility. Germline gene therapy offers a true cure for diseases, and sometimes such therapy is the only cure. Prevention is oftentimes less costly than lifetime treatment. Parents have freedom, scientists have freedom. So these are the pro arguments. The con arguments, there are scientific uncertainty and risks. It's a slippery slope to enhancement, and we don't have the consent of future generations. Allocation of resources. Germline therapy may never be cost effective. And the integrity of genetic patrimony. Future generations have the right to inherit the original genome. Last but not least is permanent versus.
This is Ted here on the question as to whether or not CRISPR editing of the human genome, especially germline modification, ought to proceed or not. I have recommended the precautionary principle, and here's an example of it. We borrow the precautionary principle from ecology and see whether or not it might apply to genetic ethics. When an activity raises threats of harm to human health or the environment, precautionary measures should be taken, even if some cause and effect relationships are not fully established scientifically. In this context, the proponent of the process or product rather than the public should bear the burden of proof, which means that a geneticist who wants to edit the human germline would have to anticipate its effects before proceeding. So I say proceed with human germline editing with caution, yes, but not yet. At this point, Arvin disagrees with me. Rather than invoke the precautionary principle, Arvin is going to suggest reversibility. That is to say, the CRISPR gene editor should proceed, but when in doubt, proceed only with reversible changes.
Reversible. In the mouse models that we have right now, mice can be modified such that they can turn on the CRISPR modification, and then we can turn it off should anything happen. This gives some flexibility that we can experiment and study some consequences and revert back if we so choose to do so, if we change our mind. I think this would be a model that we need to take into consideration seriously when applying CRISPR modifications in the future. And this is definitely already done in animal organism models. Concluding remarks. CRISPR applications are very broad. CRISPR dangers include off-target, on-target, and epigenetic effects. CRISPR regulations are unclear on where to draw the line. Right now, they draw the line between somatic versus germline and therapy versus enhancement. However, the other lines we really need to consider both ethically, philosophically, and theologically. Thank you. Most of this work are done in the context of conversations with various nonprofit organizations such as IRAS, Sinai and Synapses, and my colleagues at Harvard, GTU, and Stanford. Thank you very much.
Thank you, Arvin, for providing the science of CRISPR gene editing and labeling what could become then theological issues and ethical issues. When it comes to theology, anthropology comes to the fore, but so also does eschatology. Do we have a human nature that could be modified through gene editing? Does the human race go back to its archonic foundations, which defines us as to who we are? And then are we betraying nature if we engineer a different genome for ourselves or our children or grandchildren? I think the answer is no. The human genome has been evolving for centuries, millennia, if not years numbered in the millions. The human genome is changing right now as we speak. Well, then, should we get into our DNA with wrenches and screwdrivers and design the next generation of human beings? Why not? Well, the moral caution is, what if we make a big mistake? Or what if our prurient, selfish, self-aggrandizing, and unrealistic values are too influential on the kinds of genetic changes we make? One thing about gene alteration by engineering babies or engineering future generations is that we will realize how much in continuity with the rest of the natural world we are. DNA is DNA is DNA, and we share it with the trees, the insects, and the snakes. We will have to appreciate holistically our relationship to the rest of the world, relationally, temporally, and even eschatologically. I think the Christian theologian wants to say that the future human will be transformed. Oh, yes, there will be continuity, but there's going to be discontinuity. So we have no theological obligation to keep things the same or to return to the past. Ethics. I think it's wonderful that the scientists among us are already ethically sensitive and willing to formulate ethical issues surrounding the frontier of research and the medical applications of this CRISPR technology. Somatic cell therapy, helping people who are already living to get healing for monogenic diseases or other diseases. Is that bioethically approvable? You betcha, yippee. If we can help the health and well-being of living people through gene modification, go for it. I don't know anybody who opposes it. Germline modification, well, there we have lots of caution already, and you've heard me invoke the cautionary principle, which says we need to know what the unintended consequences are going to be before we proceed. Arvin disagrees. He says if we make only reversible changes and then we find out we made a mistake, well, then we can go back to the genome we previously had. Gene drive. I would love it if mosquitoes would stop carrying malaria. But if we're going to change the gene on the mosquito genome or make those mosquitoes go extinct, maybe we'll upset the biosphere. Maybe frogs won't get their lunch. I say proceed with caution. Arvin might say, go ahead. If we find out we made a mistake, we'll reverse it. Technological utopia. There are those in our society who believe that through technological innovation, we will so transform the human race that we will attain utopia, the perfect life. Progress will get us there. As a theologian, I have to be just a little bit more realistic and say, you know what? I don't think you can genetically modify the human genome to get rid of sin. There's no gene for sin, at least that I know of. No, utopia is not achievable through any kind of technological progress, even biotechnological progress. Finally, playing God. Should we be afraid of the bugaboo and leave our wrenches and our screwdrivers away from the DNA? No, I don't think we should be frightened by the anti-playing God rhetoric. Rather, we should think with Philip Hefner, the theologian, that the human being is the created co-creator. We are given a responsibility by God to be creative, and genetics is one of the ways in which we're going to be creative. But, of course, we have to lift up the ethical ideal of the kingdom of God, creative in ways that are directed toward healing, well-being, and as St. Anselm would say, human felicity. This is where theological anthropology, eschatology, and ethics all converge. The human being has a future. That future will be in continuity with the past and the present, but it will also engage transformation, renewal. We will pass from this creation to the new creation. As Jesus rose from the dead on the first Easter, so also will God raise us at the advent of the kingdom of God. The prophet Isaiah says, I'm about to create a new heaven and a new earth. No more shall there be in it an infant that lives but a few days, or an old person who does not live out a lifetime. Or in the book of Revelation, there will be no more crying or pain. Genetic laboratory science translated into medical therapies that bring healing and reduce crying and pain. They concretely anticipate proleptically the coming kingdom of God. Tomorrow will be different from today. Changing things in light of God's tomorrow, even changing the human genome accordingly, it's a good thing to do. Bye-bye.